The harvest operation follows the production cell culture operation. Its purpose is to remove non-dissolved solids from the process stream to prepare the process stream for purification steps such as chromatography. To start with, the process is at the end of its cell culture run inside the bioreactor, in this case a 6,000-liter stainless steel bioreactor controlled by the Distributed Control System, or DCS. The first unit operation in this harvest sequence is centrifugation. The disc stack centrifuge from Alpha Laval spins at thousands of revolutions per minute to generate a separating force thousands of times the force of gravity. As it spins, the feed stream from the bioreactor is continuously pumped in, and most of the solids are separated into a heavy phase that continuously flows to waste. The centrifuge is controlled by a programmable logic controller. The light phase flowing from the centrifuge moves on to the next unit operation, depth filtration. In this case, a pod single-use system from Millipore Sigma is used. Unlike the centrifuge, this unit operation has no parts moving during the separation. The process stream passes straight through the filters, leaving behind more of the undissolved solids. Following depth filtration, the process stream leaves this area by pipe and passes through absolute filters, not shown, to complete its clarification. After passing through the absolute filters, the clarified process stream passes into the harvest tank, in this case a 6,000-liter stainless steel tank controlled by the DCS. Now the material is ready for capture.